Hi, I'm Judy Cole, the Executive Vice President and CEO of the MIT Alumni Association, and I'm delighted to welcome you to this web production of the MIT Alumni Association.
Please welcome to the stage Ron Weiss.
Thank you. Pleasure to be here this morning. I've been giving instructions that you have to do your questions at pollev.com/MITQ. You are not allowed to raise your hands. Is that right? Because that's what I've been told. So use technology and internet and all that. Okay, so it's a pleasure to be here. I'm actually a computer scientist by training. And about 20 years ago, when I was a PhD student here in Core 6, I became fascinated with the notion that we might be able to program cells in the same way that we program computers. And I've been essentially doing that ever since and kind of become addicted to this notion of trying to program cells. So now that field is essentially called synthetic biology. What is it that we're trying to do? So what we do is we come up with some kind of high-level behavior that we want the cells to be able to execute. Then we write that high-level behavior as a biological program. So essentially, we write code, but we don't put that code into computers. We want to put that code into living organisms. So we take that high-level code that describes a particular behavior, and then we convert that to some kind of a gene regulatory network. We then take that gene regulatory network and figure out how to actually encode that using a DNA molecule. And with that DNA molecule, we synthesize that in the lab and end up putting that inside cells as a way of extending or modifying the behavior of cells. And so what we're trying to do in synthetic biology is understand how to develop an engineering discipline that will allow us to do that reliably, predictably, efficiently, and so on. And so as a young computer scientist here, I wanted to think about cells as being these nice modular entities that we can basically do plug-and-play operations just with DNA. So I was maybe a little bit naive at the time. And as I've kind of gotten older, more gray hair, I guess more male pattern baldness, realized that I should probably respect and appreciate biology a little bit more. So maybe think about biology kind of in a different sense. It's not quite as clean as we'd like it to be. So I would say I'm not quite at the notion of thinking that it's too messy. I'm probably in the notion that it is modular, it is programmable, but we need to really appreciate the subtleties in the biological substrate. And so we need to understand what are the things that make it challenging to genetically engineer cells to do exactly what we want. So for example, the notion of gene expression noise, biological entities are very noisy. How can we create reliable behavior in these very noisy substrates? And so for that, we're working on combining a systems and synthetic biology approach to be able to engineer these cells robustly and predictably. So what we do is we work on the foundational aspects of synthetic biology. How can we program cells? But we also are trying to understand where would that matter, okay? And so I'll talk about a couple of examples where we believe the notion of being able to program cells somehow fundamentally changes the approach, somehow fundamentally changes the way we approach things. And so one of the examples I'm going to talk about is cancer therapy, where the notion is we're building programs that can very specifically distinguish between cancer and non-cancer cells and then execute a therapy. And the other one that I'm going to talk about, and that relates to what Linda mentioned, was this notion of programmable organoids. How can we use synthetic biology and genetic engineering to create any kind of organoid that we want? Okay, so I'll start off with cancer. So this is what we'd like to be able to do for therapy for cancer. So what we're doing is incorporating essentially biocomputers inside engineered viruses, okay? And so the way this starts, that we can think about this as a multi-step program, where the initial step is we take this virus and we infect the virus into somebody patient that actually has a tumor. And the notion is that the virus will go both into healthy cells and tumor cells. And importantly, the first thing that it does is it computes whether the particular cell that it infected is a cancer cell or a healthy cell. And if it's a cancer cell, then it begins to do selective replication. So it will selectively replicate to the tumor cells, whereas in the healthy cells, it basically does nothing and eventually just goes away. Once it did this selective spreading, it will begin to execute cancer death only in these tumor cells, and that's the thing you see here. Oftentimes, the virus won't get into every single cell, and so as a result of that, the next thing it would do is try to recruit the immune system to help out and kill the rest and the remaining cells they wasn't able to access. And so, for example, it would get T cells to come in. They would help actually kill the tumor cells that were not directly infected. And eventually, we would get a situation where hopefully the tumor cells will actually go away, and then the healthy cells will remain. And so the key here is that we need to have a mechanism very precisely allows the virus to determine, is this a cancer or a non-cancer cell? And so for that, we have the notion of going into a cell. The virus goes into a cell and inspects biomarkers inside the cell. Okay, so it actually tries to sense what biomarkers are present in this particular cell, and do they match a profile of biomarkers that's indicative of a tumor cell. So for example, what you're seeing here are tumor cells that have biomarkers A and B, and other cells have biomarkers as well, A and C and B, but no cell has both A and B present in it. Okay, so there's this notion that if you're able to look at enough biomarkers, one is not enough, but if you're able to have the virus investigate enough biomarkers, it can make a very specific decision about whether this cell is a tumor cell or not. So in this particular case, it would be a simple, quote unquote, simple logic function. If A and B are high, then it's a tumor cell. You can have heterogeneous tumors. In this case, basically what you do, so in this case, the rest of this population would have not just A and B high, but maybe B and E high. And so for that, you can develop a more complex logic function that is using an OR operation. So if A and B are high, or E and B are high. So we've been able to actually demonstrate the ability to do AND logic, OR logic, and NOT logic. So let me actually tell you a little bit on how this works. So imagine that we have a particular cancer, such as HeLa cervical cancer, and it has the following biomarkers where miR-141, this is microRNA 141, is low, and miR-21 is high, okay? And so that somehow, those biomarkers are present in HeLa cells. And so you can think about trying to cure or address this situation with a logic function where it says if miR-21 is high and miR-141 is not high, then execute a therapy. So this is a simple logic function, and the question is, can we actually implement this using DNA? Okay, so the way we would do that is with this particular construct. Okay, so this has the following features. I won't get into this in complete detail, but it has the following features, which is this arrow that says make a protein, this inverted T, which says basically repress, and the production of a killer protein. Okay, so these are the essential elements here, and it says that if microRNA 141 is high, then it represses the production of the killer protein. Okay? And then if microRNA 21 is high, it represses the repressor of the killer protein. Okay? So basically, this is acting like a not operation. So the only way that we have production of the killer protein is that if this is low and this is high. So this is essentially a simplified genetic circuit that activates only when miR-141 is low and miR-21 is high. Okay. So we focused initially on HeLa cervical cancer. This was published as a New York Times bestseller about it, recent movie. It's a really interesting story about this. I won't get into the societal aspects of this cell line, but this is the most common cell line in the world. And we did some bioinformatics, and we realized that in order to be able to distinguish HeLa versus all other cell types, you can't do it with 2 biomarkers, but we can do it with 6. So in particular, these 3 biomarkers have to be low, and so what they do is they repress the production of the output protein, and then this microRNA 21 has to be high, and basically, again, it's doing the not operation, and also these combination of these microRNAs has to be high as well. So you're seeing here, without, again, going into all the details, that there's a logic function that we'd like to satisfy, and here's a genetic circuit that is able to implement that logic function. So we went ahead and implemented that, and we're able to demonstrate experimentally that HeLa cells, when they're when this DNA is introduced into them, the output is high, whereas other cell types, the output is low. Okay? So this worked reasonably well. but it wasn't quite perfect. So we're able to, what we're seeing here is about a 3-4-fold difference between HeLa high output and the low output that we are seeing in non-HeLa cells. Okay? We looked at single cell behavior. We also were able to demonstrate that even with this 3-4-fold difference, we're able to get selective killing such that the circuit kills mostly HeLa cells and doesn't harm the other cells. But we didn't feel this was quite enough for our purposes to go into actual therapy. And so we spent about 5 years, so this is work by a postdoc in my lab, taking this and trying to optimize it. So he started with this simplified gene regulatory network, and he put a feed-forward motif, and another feed-forward motif, and another feed-forward motif, and a feedback motif, and another feedback motif, and a feed-forward motif. Anyways, I'm not going to explain all the details about this, but this is actually even a simplified version of this. This is what it really looks like. So you see it's really messy. It kind of looks like, you know, a messed up map of the T or something like that when it's under construction, right? But this is the genetic circuit that we built. So it's very complex, but it's also beginning to look like, rather than a simplified Boolean logic function, it begins to actually look like a biological network, a natural biological network. And so we're trying to understand, what are the design principles? Why, how does this actually relate to the natural designs that biology is actually engineered? Maybe there's a reason biology engineers things in a messy way, not to make our lives easier, but maybe to give it the right kind of robustness and performance. And indeed, once we were able to implement this, we went from this 3-4-fold on-off ratio to over 100-fold on and off. So we got the performance we need from this kind of messy implementation. Okay, and so these are very preliminary results, and I guess we're excited about it that we are trying to cure cancer in mice. We're still excited about this. But the notion here is that we have breast cancer, and we infect the virus into breast cancer in mice, and we see that the negative control, when the virus just replicates, it goes all over the mouse. And then you see here on the very right side, when we have this optimized virus that is able to make a very precise distinction between the breast cancer tumor and the healthy cells, you see that the virus basically just remains in the breast cancer. So we're pretty excited about this notion. Let me, and we're trying to begin to think about how to potentially move this to clinical trials. Let me very quickly talk about another vignette which relates to what Linda was talking about with respect to organoids. So the question is, how can we use synthetic biology? How can we use the notion of programming to create organs by design? Okay, any kind of organ that we want. And so the notion is, can we take embryonic stem cells or these induced pluripotency stem cells that are patient-specific and then get them to proliferate and then create these two-dimensional patterns which then end up differentiating into a collection of cells that ends up creating an organoid by design. And so what we want to do this, and this again relates to what Linda was mentioning, we want to ultimately use this to do drug development. So for example, wouldn't it be nice if we had the ability to create a liver organoid that would be genetically identical to you, be made out of your skin cells, where we would test the drugs on that liver organoid as opposed to some kind of random mouse or rat that's not as related to you. Okay? We ultimately actually want to also be able to build construct liver organoids that are large enough that to think about doing transplantation with them for organ regeneration. So here's the general thing that we'd like to be able to do with synthetic biology is write multi-step biological programs that take these embryonic stem cells and then step them through essentially mimicking embryogenesis. So get them to differentiate in multiple steps until they reach the cell type that we want. So we think about this by writing these biological programs that are essentially using if-then statement. If this is this cell type, then begin to differentiate to this cell type. If this is a cell type, then begin to differentiate more and more and more and more. Okay? And so let me show you a quick example of one of those. Again, this was in collaboration with Linda, postdoc in her lab, Mo, and postdoc in my lab, Patrick. And we asked the question, can we begin to make these kind of organoids? And the first thing we wanted to do is, can we take these iPS cells and make endoderm out of them? Okay? And so we found that this special transcription factor called GATA6 is actually very powerful. Okay? Specifically, we genetically engineered iPS cells to make GATA6, but at very different levels. Okay, so there's a variety of different GATA6 expression levels. Okay? And what happened is, this is day 0. When we look at it, there's basically no pattern. But after 5 days of expression of GATA6 at different levels, the cells begin to self-assemble and then create all these structures. Basically, we have symmetry breaking. And it relates to the level of GATA6 in the system. So if there's high GATA6 in the cell, it becomes endoderm. If there's low GATA6, it remains pluripotent. And if there's medium GATA6, what it does, it depends on its neighbors. So they're kind of acting in a social way, if you will. Okay, so after that, so we now have the ability to express GATA6 at different levels. High GATA6 results in endoderm and also mesoderm. The low GATA6, they end up becoming ectoderm. So we have a mechanism now in a very controlled way. And this is, Linda was mentioning the notion of reliability. This is essentially repeatable every single time. We can dial in and we say, we want this percentage of endoderm, we want this percentage of ectoderm. We get it essentially every single time. And then we can let the system continue to develop. And from that, we're actually able to generate, so as I mentioned, endoderm, mesoderm, and ectoderm, which basically are the germ layers that give rise to every single cell type in your body. And if we wait long enough, the endoderm and the mesoderm develop into a liver bud, an embryonic liver, and the ectoderm develops into brain. So we have basically an organoid that's a liver-brain organoid that is basically patient-derived. Excuse me. And so we can look at this. This is endoderm. We can stay endoderm, ectoderm. And the ectoderm ends up becoming this forebrain structure. So we grow this initially in a 2D layer. And then from the 2D layer, we begin to see rise of this kind of 3-dimensional forebrain. So we have this kind of possibly smart organoid that's being grown on a chip. And so that got us thinking, can we go beyond 2D? Can we begin to think about 3D? And so a graduate student in my lab, Patrick, began to grow these cells in these U-bottom wells. And so these cells end up collecting themselves, aggregating into spheres. And so this is what you're seeing here after day 12. And these spheres are highly vascularized. Okay, so what you're seeing here is vascularization. There's basically intense vascularization that ultimately allows them to grow and grow and grow and grow. So this is one of the big problems with organoids, is that liver organoids, for example, is that they can't grow more than 1 millimeter in thickness, because once they do that, the inner cells do not have access to nutrients and they die. But once you have this kind of intense vascularization, the organoid is able to grow and grow and grow. And if you wait long enough, 74 days in this particular case, they become basically huge. And so they become multiple centimeters in diameter. We're calling them meganoids. And you maybe play hacky sack with them. They're really quite large. This is a zoom in of one of these liver organoids that you're seeing here. So you really can begin to think about possible transplantation of these organoids. And the very last thing I want to mention is that now that we have the ability to create 3-dimensional shapes, can we possibly program these 3-dimensional shapes? And so very briefly, what we want to do is be able to genetically control cadherin expression. Cadherin are these molecules that result in cell adhesion. Okay, and so I'll just show you quick movies about that. So when we have cells that have high adhesion, and that's going to be the red cells, and cells that have low adhesion, we put them together, and you see that they kind of begin to aggregate, and the high adhesion cells remain in the middle and ball up, and the cells that are low adhesion kind of remain on the outside. And we can do other cells where just mostly high adhesion cells, and you see that they begin to ball up really quite nicely, and the low adhesion cells are on the surface. And then when we have fewer high adhesion cells, they actually make, this was slightly surprising to us, they make all these clusters of high adhesion cells that are connected by low adhesion cells. Right? So we now have beginning ability to create 3-dimensional structures based on cell-cell interactions. Okay, and so just to sum up, in the cancer work, we believe that this kind of notion of complex, messy design is the way to create robust systems. And not just think about that in terms of like the simplified Boolean functions. And then in the organoids, I showed you that this notion of noise, where different cells are making GATA6 at different levels, actually was responsible for the creation of an actual organoid. And with that, I showed folks from my lab that were responsible for this work, and I'll stop by thanking you.
Thanks again for joining us. For more information on future MIT Alumni Association productions, please visit our website.